Today, we'll be doing a mind map on cell division. There are three types of cell division: mitosis, amitosis, commonly referred to as binary fission, and meiosis. Out of the three, mitosis and meiosis will be the common ones you come across. Firstly, we have mitosis. Mitosis is a form of nuclear division that produces daughter nuclei containing the same number of chromosomes as that of the parent. What this means is that when a cell divides into two, the nucleus of the parent cell divides so that both daughter nuclei are genetically identical to that of the parent. There are three main reasons why a cell needs to undergo mitosis. The first is growth, where an increase in organism size has to be accompanied by cell division and differentiation to form tissues and organs. The second reason is to repair worn-out parts or to replace old and dead cells. One common example of this is during injury. The last reason is for asexual reproduction, as is the case for plants and microorganisms. And this brings me to amitosis, which is a form of asexual reproduction common for the reproduction of prokaryotes, for example bacteria, and some eukaryotes as well. I'll come back to how this takes place later on. To fully understand cell division, we must first understand the cell cycle. The cell cycle has two main phases: interphase and cell division. But this can be further split into 6 sections: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Mitosis actually only forms a small part of the entire cell cycle, which is shown here. At any one point in time, a cell will be somewhere in this cycle. Interphase itself consists of 3 phases: G1, S, and G2 in that order. Between each of these stages is essentially a checkpoint as the cell prepares for cell division. The cell's movement from one stage of the cycle to the next is controlled by proteins called cyclins. How they work is that they bind to cyclin-dependent kinases, which is an enzyme which triggers a chain reaction, leading to the cell carrying out a task specific to a phase in the cell cycle. This is why interphase takes so long, taking up over 3/4 of the entire cell cycle. There are 3 main things that the cell carries out in interphase to prepare for mitosis. First, it stores up energy in the form of ATP for cell division later on. Secondly, it replicates organelles and centrioles. It replicates organelles as the cell will be undergoing mitosis to form 2 daughter cells, and each daughter cell will need its own set of organelles. Centrioles are replicated as they are essential for mitosis, which will be seen later on. Lastly, replication of chromatin DNA occurs. For every chromatin DNA strand, it will be replicated to form sister chromatids, where both are identical DNA molecules. The replication occurs about the centromere, which is a specialized protein, and there is only one of it per chromatin thread. This DNA replication is done using RNA. This is something that will be covered in my mind map on DNA replication. The importance of this DNA replication is to be able to produce 2 daughter nuclei which are genetically identical. Okay, so now back to mitosis. As I mentioned, in the entire cell cycle, mitosis is only a small part of it, starting with prophase and ending at telophase. Do note that cytokinesis is not considered part of mitosis, where it's only the splitting of the cell to form 2 independent daughter cells. Mitosis begins firstly with prophase. In prophase, chromatin threads in the nucleus condense. They coil and shorten to become chromosomes. This process is referred to as supercoiling. At the same time, esters begin forming around the previously mentioned centrioles, and the 2 pairs of centrioles begin to move to opposite poles of the cell. The nucleolus is responsible for production of rRNA, which occurs during interphase to allow for DNA replication. The nucleolus is disassembled during cell division, and the nuclear envelope begins to disintegrate as well. Esters around centrioles begin synthesizing spindle fibers, which extend and connect to the centromeres of the chromosomes. The next stage of mitosis is metaphase, where the chromosomes line up along the equatorial plane of the spindle. The spindle fibers attached to the centromeres are adjusted accordingly to line the chromosomes up. One thing to note in the diagram is that the centromere is attached to spindle fibers on both sides. This is because the chromosomes will be pulled apart in the next stage of anaphase. In anaphase, the centromeres divide and spindle fibers shorten to pull the split chromatids to opposite poles of the cell. One interesting thing is that at this point, there are double the number of chromosomes in the cell, as one centromere equates to one chromosome, if you were to remember. Once the chromosomes have been pulled to the opposite poles of the cell, the spindle fibers break down and the nuclear envelope reforms around the two separate regions of DNA. The nucleolus also reforms and the chromosomes uncurl to form chromatin threads. For a short while, the cell will have two nuclei. The last stage of cell division is cytokinesis, where the cytoplasm divides. Contractile rings cause furrows to form, where two opposite ends of the cell membrane come together to be pinched into two independent daughter cells. So now coming back to binary fission. In binary fission, the sole strand of circular DNA in bacteria, for instance, replicates to form two distinct regions of DNA. As this occurs, the membrane of the cell elongates and furrows begin to form as the cell begins to divide, just as it does for cytokinesis after mitosis. When opposite ends of the membrane come together, the whole cell pinches off to form two daughter cells. So now we have talked about mitosis and binary fission. Before we go into meiosis, you may want to review the process of mitosis because the mechanisms are very similar to that of meiosis. Meiosis is a form of nuclear division that produces daughter nuclei with half the number of chromosomes as that of the parent. Note this key difference with mitosis. The reason for the production of haploid cells is for the formation of gametes, also known as sex cells, and logically, this is for the purposes of reproduction via fertilization. For every diploid cell that undergoes meiosis, it produces a total of 4 gametes, each which are haploid in nature, meaning it has only half the number of chromosomes as that of the parent cell. Meiosis is only restricted to germ cells, found in the testes and ovaries for males and females respectively. Before a cell undergoes meiosis, it goes through the same stages of the cell cycle before beginning meiosis. In meiosis, the cell goes through 2 rounds of division, as you will see. Meiosis I begins with Prophase 1, where the same things happen as that in mitosis. Chromosomes are formed and esters form around centrioles that begin moving to opposite poles of the cell. The nucleolus and nuclear envelope also begin disintegrating towards the later part of Prophase 1. The difference is that in Meiosis I, homologous chromosomes are paired up, which is a pair of matching chromosomes. In humans who have a total of 46 chromosomes, there will be a total of 23 pairs of homologous chromosomes that form. As homologous chromosomes pair up, the chromatids may coil and twist around each other. The strength of this coiling may cause crossing over, which produces new alleles along the chromosomes. This is how genetic variation comes about. This point where the chromosomes cross is called chiasma, and crossing over can occur along the whole length of the chromosome and in every 23 pairs of homologous chromosomes. In mitosis, spindle fibers from opposite poles of the cell attach to the same centromere. The difference here is that spindle fibers from opposite poles of the cell attach to the centromeres of either homologous chromosomes. This is to split the homologous chromosomes up later on. Once the spindle fibers are attached to the centromeres, they are lined up along the equatorial plane of the spindle in Metaphase 1. Following this, the cell completes the remaining stages of Meiosis I, which are essentially the same as that of mitosis. The homologous chromosomes with their new alleles are split up to opposite ends of the cell, and 2 distinct regions of DNA are formed. After going through cytokinesis 1, the cell splits up into 2 daughter cells, whose daughter nuclei are already haploid. The 2 daughter cells produced from Meiosis I then go on to a second round of cell division called Meiosis II. In Meiosis II, the mechanisms are even more similar to that of mitosis, where in Prophase II, spindle fibers are once again synthesized which are attached to centromeres of the chromosomes. The chromosomes are lined up once again in Metaphase II and get split into 2 in Anaphase II. The split chromosomes are put to opposite ends of the cell to form 2 separate nuclei. Both cells go through cytokinesis 2 to form a total of 4 daughter cells, each with half the number of chromosomes as that of the parent diploid cell. As seen in the daughter cells produced, there are different variations in the nucleus. These daughter cells are now gametes, where separate haploid gametes from males and females can merge together to form a diploid cell called a zygote, through fertilization to form a new organism. This process will be covered in my mind map on sexual reproduction. In exams, mitosis and meiosis are the main types of cell division you will need to know, especially in how the two differ. Some key differences are: firstly, there is only one nuclear division in mitosis, while there are two in meiosis, meiosis I and II. Secondly, mitosis produces daughter cells with the same number of chromosomes as the parent, while meiosis produces haploid cells. Thirdly, the daughter cells produced by mitosis are genetically identical to the parent, while it is not so for meiosis due to things such as crossing over. Fourthly, mitosis occurs in cells all over the body, while meiosis only occurs in gonads, or sex cells. Fifth difference is that mitosis produces 2 daughter cells, while meiosis produces 4 daughter cells. Lastly, crossing over and pairing of homologous chromosomes do not occur in mitosis, but do so in meiosis. A comparison of the differences can come out in the form of an essay question, so do remember a couple of these differences. So this is the end of my mind map on cell division. Hope you guys find it useful and have a good day.